ये है रिस्पिरेशन इन द प्लांट्स का एनसीईआरटी। तो एनसीईआरटी कहता है कि ऑल ऑफ अस ब्रीथ टू लिव, बट व्हाई ब्रीथिंग सो एसेंशियल टू लाइफ? What happens when we breathe? Also, do all the living organisms, including plants and the microbes, breathe? If so, how? All living organisms need energy for carrying out their daily life activities, be it absorption, transport, movement, reproduction, or even breathing. Where does all this energy come from? We know we eat food for the energy, but how is this energy taken from the food? How is this energy utilized? Do all food give the same amount of energy? Do plants eat? Where do plants get their energy from? And microorganisms for their energy requirement, do they eat food? कहता है कि you may wonder at the several questions laid above. They may seem to be very disconnected, but in reality, the process of the breathing is very much connected to the process of release of the energy from the food. Let us try and understand how this happens. All the energy required for the life process is obtained by the oxidation of some macromolecules that we call the food. Only green plants and the cyanobacteria can prepare their own food by the process of the photosynthesis. They trap the light energy and convert it into the chemical energy that is stored in the bonds of the carbohydrate like the glucose, sucrose and the starch. We must remember that in the green plants too, not all cells, tissue and the organs photosynthesize. Only cells containing the chloroplast that are most often located in the superficial layers carry out the photosynthesis. Hence, even in all green plants, all other organs, tissue and the cells that are non-green need food for the oxidation. Hence, food has to be translocated to all the non-green parts. Animals are the heterotrophic, that means they obtain food from the plants. Directly herbivores or the indirectly carnivore. Saprophytes like the fungi are dependent on the dead and the decaying matter. What is important to recognize is that ultimately all the food that is required for the life process comes from the photosynthesis. This chapter deals with the cell respiration or the mechanism of the breakdown of the food materials within the cell to release the energy and trapping of this energy for the synthesis of the ATP. Photosynthesis of course takes place within the chloroplast in the eukaryotes, whereas the breakdown of the complex molecule to yield the energy takes place in the cytoplasm and in the mitochondria, also only in the eukaryotes. The breaking of the Phi bond of the complex compounds through the oxidation within the cells leading to the release of the considerable amount of the energy is called the respiration. The compounds that are oxidized during this process are known as the respiratory substrate. Usually carbohydrates are oxidized to the release energy, but proteins, fats and or even the organic acids can be utilized can be used as a respiratory substrate in some plants under the certain conditions. During the oxidation within the cell, all the energy contained in the respiratory substrate is not released free into the cell or in a single step. It is led in a series of the slow stepwise reaction controlled by the enzymes, and it is trapped as the chemical energy in the form of the ATP. Hence, it is important to understand that the energy released by the oxidation in the respiration is not, or rather cannot be used directly, but it is used to synthesize the ATP, which is broken down whenever and wherever energy needs to be utilized. Hence, ATP acts as the energy currency of the cell. This energy trapped in the ATP is utilized in the various energy requiring process of the organism, and the carbon skeleton produced during the respiration is used as a precursor for the biosynthesis of the other molecules in the cell. फिर कहता है कि do plants breathe? So, well, the answer to this question is quite not so direct. Yes, the plant require the O2 for the respiration to occur, and they give out the CO2. Hence, plants have the system in the place that ensure the ability of the O2. Plant, unlike the animals, have no specialized organs for the gaseous exchange, but they have the stomata and the lenticels for this purpose. There are several reasons why plants can get along without the respiratory organs. First, each plant takes part care of its own gaseous exchange. There is very little transport of the gases from the one part of the plant to the another. Second, plants do not present get demands for the gas exchange. Root, stem and the leaves respire at the rates far lower than the animals do. Only during the photosynthesis are the large volume of the gases exchanged, and each leaf is well adapted to take care of its own needs during these periods. When often when cell photosynthesis ability of the O2 is not a problem in this cell, since O2 is lived within the cell. Third, the distance that the gases must diffuse even in a large bulky plant is not great. Each living cell each living cell in a plant is located quite close to the surface of the plant. This is true for the leaves. You may ask, but what the thick woody stem and the root and the Roots. In the stem, the living cells are arranged in the thin layer inside and beneath the bark. They also have opening called the lenticels. The cells in the interior are dead and provide only mechanical support. Thus, the most cells of the plant have at least a part of their surface in contact with the air. This is also facilitated by the loose packing of the parenchyma cells in the leaves, stem, and the roots, which provide an interconnected network of the air passage. The complete combustion of the glucose before the CO2 and the H2O as the end products yields the energy, most of which is given out as the CO2. तो यहाँ पर आता है 6 6 प्लस 6 CO2 देता है 6 CO2 प्लस 6 H2O प्लस दी एनर्जी है जो देता है. फिर कहता है कि if this energy is to be useful to the cell, it should be able to utilize it to synthesize other molecules that the cells require. The strategy that the plant cell uses it to catabolize the glucose molecule in such a way that not all the liberated energy goes out as a heat. The key to oxidize glucose not in one step but in several small steps, enabling some steps to be just large enough such that the energy released can be coupled to the ATP synthesis. How this is done is essentially the story of the respiration. फिर कहता है कि during this process of the respiration, oxygen is utilized and the carbon dioxide, water and the energy are relieved as the products. The combustion reaction requires the oxygen, but both some cells live where oxygen may or may not be available. Can you think of such situation and the organism where O2 is not available? There are sufficient reasons to believe that the first cell on this planet lived in the atmosphere that lacked oxygen. Even among the present day living organisms, we know of the several that are adapted to the anaerobic conditions. Some of these organisms are facultative anaerobes, while in others the requirement for the anaerobic condition is obligate. In any case, all living organisms retain the enzymatic machinery to partially oxidize glucose without the help of the oxygen. This breakdown of the glucose to the pyruvic acid is called as the glycolysis. Now the term glycolysis is originated from the Greek word glykos for the sugar and the lysis for the splitting. The theme of the glycolysis was given by the Gustav Embeden, Otto Meyerhoff and the Jeff Parnas, and is often referred to as the EMP pathway. In the anaerobic organism, it is the only process in the respiration. Glycolysis occurs in the cytoplasm of the cell and is present in all living organisms. In this process, glucose undergoes partial oxidation to form the two molecules of the pyruvic acid. In plant, this glucose is derived from the sucrose, which is the end product of the photosynthesis, or from the storage. Carbohydrate. Sucrose is converted into the glucose and the fructose by the enzyme invertase, and these two monosaccharides readily enter the glycolytic pathway. Glucose and the fructose are the phosphorylated to give rise to the glucose 6-phosphate by the activity of the enzyme hexokinase. This phosphorylated form of the glucose then isomerized to produce the fructose 6-phosphate. Subsequent steps of the metabolism of the glucose and the fructose are the same. The various steps of the glycolysis are depicted in table 14.1. In glycolysis, a chain of the 10 reactions under the control of the different enzymes take place to produce the pyruvate from the glucose. While studying the steps of the glycolysis, please note the steps at which utilization of the synthesis of the ATP, or in this case NADH plus H plus, take place. फिर कहता है कि ATP is utilized at the two steps, first in the conversion of the glucose into the glucose 6-phosphate and second in the conversion of the fructose to the fructose 6-phosphate to the fructose 1,6-biphosphate. The fructose 1,6-biphosphate is split into dihydroxyacetone phosphate and the 3-phosphoglyceraldehyde. We find that there is one step where NADH plus H plus is formed from the NAD plus. This is when the 3-phosphoglyceraldehyde is converted to 1,3-bisphosphoglycerate. Two redox equivalents are removed in the form of the two hydrogen atoms from the PGL and transferred to a molecule of the NAD+. PGL is oxidized and with the inorganic phosphate to get converted into the BPGA. The conversion of the BPGA to the 3-phosphoglyceric acid is also an energy yielding process. This energy is trapped by the formation of the ATP. Another ATP is synthesized during the conversion of the PEP to the pyruvic acid. Can you then calculate how many ATP molecules are directly synthesized in this pathway from the one glucose molecule? फिर आता है कि pyruvic acid then the key product of the glycolysis. What is the metabolic phase of the pyruvate? This depends on the cellular need. There are three major ways in which different cells can handle pyruvic acid produced by the glycolysis. These are the lactic acid fermentation, alcoholic fermentation, and the aerobic respiration. Fermentation takes place under the anaerobic condition in many prokaryotes and unicellular eukaryotes for the complete oxidation of the glucose to the CO2 and the H2O. However, organisms adopt the Krebs cycle, which is also called the aerobic respiration. This requires the O2 supply. तो स्टेप्स ऑफ द ग्लाइकोलाइसिस में किस प्रकार से है, तो उसमें दिया हुआ है सबसे पहले ग्लूकोज को एटीपी ग्लूकोज को जो कि 6 कार्बन का है उसमें एक एटीपी को खर्च किए और ग्लूकोज 6 फॉस्फेट में कन्वर्ट किए। ग्लूकोज 6 फॉस्फेट को आइसोमरेज के द्वारा फ्रुक्टोज 6 फॉस्फेट में कन्वर्ट किए। फिर एक एटीपी का खर्च किए और फ्रुक्टोज 1,6-bis फॉस्फेट में कन्वर्ट किए। फिर फ्रुक्टोज 1,6-bis फॉस्फेट को एल्डोलेज के द्वारा ट्राइएज फॉस्फेट के 2 मॉलिक्यूल एक पीजीएल और एक ढाप में तोड़ दिए। फिर पीजीएल और ढाप को आइसोमरेज के द्वारा एक दूसरे में कन्वर्ट कर दिए और फिर है जो 1,3-बाइफॉस्फोग्लिसरिक एसिड बना दिए। 1,3-बाइफॉस्फोग्लिसरिक बनाते समय NAD+ है जो NADH में कन्वर्ट हो गया। आगे फिर साइकिल इधर से जो साइकिल जो है ये 2 टाइम्स चल रहा है। उसके बाद फिर आता है कि 1,3-बिस-फॉस्फोग्लिसरिक एसिड को 3-फॉस्फोग्लिसरिक एसिड में कन्वर्ट कर दिए जिसमें हमको एक एटीपी बन गया। फिर 3-फॉस्फोग्लिसरिक एसिड को 2-फॉस्फोग्लिसरेट में कन्वर्ट करते हैं आइसोमरेज के द्वारा। फिर 2-फॉस्फोग्लिसरेट से एक पानी निकालकर 2-PP यानी कि फॉस्फोनोल पायरुवेट में कन्वर्ट करते हैं। फिर फॉस्फोनोल पायरुवेट से पायरुविक एसिड है जो बना जाते हैं। तो फॉस्फोनोल पायरुवेट से जब पायरुविक एसिड बना रहे थे तो उसमें भी 2 एटीपी हमको मिलता है। तो टोटल 4 एटीपी मिले, 2 एटीपी इधर जो खर्च हुआ नेट हमको 2 एटीपी मिला। फिर आता है फर्मेंटेशन। तो in the fermentation, say by the yeast, the incomplete oxidation of the glucose is achieved under the anaerobic condition by effects of the reaction where the pyruvic acid is converted to the CO2 and the ethanol. The enzymes pyruvic acid decarboxylase and alcohol dehydrogenase catalyze this reaction. Other organisms like some bacteria produce lactic acid from the pyruvic acid. The steps involved are shown in the figure 14.2 in the animal cells, also like the muscles during the exercise. When oxygen is inadequate for the slow respiration, pyruvic acid is reduced to the lactic acid by the lactate dehydrogenase. The reducing agent is NADH+H+, which is reoxidized to the NAD+ in both the processes. फिर कहता है कि in both the lactic acid and the alcohol fermentation, not much energy is released, less than 7% of the energy in the glucose is released and not all of it trapped at the high energy bonds of the ATP. Also the processes are hazardous. Either acid or the alcohol is produced. What is the net ATP that is synthesized? Calculate how many ATP are synthesized and deduct the number of the ATP utilized during the glycolysis when one molecule of the glucose is fermented to the alcohol or the lactic acid. Yeast poison themselves to death when the concentration of the alcohol reaches about 13%. What then would be the maximum concentration of the alcohol in the beverage that are naturally fermented? How do you think alcoholic beverage of the alcohol content greater than this concentration are obtained? What then is the process by which the organism can carry out complete oxidation of the glucose and extract the energy of storage too? फिर कहता है कि synthesize a larger number of ATP molecules are needed for the cellular metabolism. In the eukaryotes, these steps take within the mitochondria and this requires the O2. Aerobic respiration is the process that leads to a complete oxidation of the organic substance in the presence of the oxygen and it releases the O2, CO2, water and a large amount of the energy present in the substrate. This type of the respiration is most common in the higher organism. We will look at this process in the next section. फिर ये दिया हुआ है मेजर पाथवेज फॉर द एनारोबिक रेस्पिरेशन। इसमें क्या किया है कि ग्लूकोज को ग्लिसरॉल्डिहाइड 3-फॉस्फेट में कन्वर्ट किए हैं। फिर 3-फॉस्फोग्लिसरिक में इस समय करते समय NAD+ जो NADH में कन्वर्ट हो गया। फिर 3-फॉस्फोग्लिसरेट फॉस्फोइनोल पायरुविक एसिड बनाए और फॉस्फोइनोल पायरुविक एसिड से पायरुविक एसिड बना दिए। लैक्टिक एसिड में क्या करते हैं कि पायरुविक एसिड को हम फर्मेंटेशन में पायरुविक एसिड से इथेनॉल प्लस CO2 बना सकते हैं जिसमें NADH+H+ है जो NAD+ बनाएगा। ये NAD+ वापस ग्लाइकोलाइसिस में जाके यहाँ पर जाके यूज होगा। उसी तरह से पायरुविक एसिड को लैक्टिक एसिड में भी कन्वर्ट कर सकते हैं और NADH को NAD+ में कन्वर्ट कर सकते हैं और ये NAD+ को यहाँ ला के दोबारा से है जो यूज कर सकते हैं। फिर कहता है एरोबिक रेस्पिरेशन। तो For the aerobic respiration to take place within the mitochondria, the final product of the glycolysis, pyruvate, is then transported from the cytoplasm into the mitochondria. The crucial events in the aerobic respiration are the complete oxidation of the pyruvate by the stepwise removal of all the hydrogen atoms, leaving 3 molecules of the CO2, the passing of the electrons formed as a part of the hydrogen atom to the molecular O2 with the simultaneous synthesis of the ATP. What is interesting to note is that the first process takes place in the matrix of the mitochondria, while the second process is located on the inner membrane of the mitochondria. Pyruvate, which is formed by the glycolytic catabolism of the carbohydrates in the cytosol, after it enters the mitochondrial matrix, undergoes the oxidative decarboxylation by a complete set of the reaction catalyzed by the pyruvic dehydrogenase. The reaction catalyzed by the pyruvic dehydrogenase requires the participation of the several coenzymes, including the NAD+ and the coenzyme A. तो इसमें क्या हुआ कि पायरुविक एसिड प्लस कोएंजाइम ए पायरुविक एसिड प्लस कोएंजाइम ए है और उसमें NAD+ मिला है, तो मैग्नीशियम आयन के प्रेजेंस में और पायरुवेट डिहाइड्रोजन के प्रेजेंस में एसिटाइल CoA प्लस CO2 प्लस NADH प्लस H+ बनता है. और during this process, 2 molecules of the NADH are produced from the metabolism of the 2 molecules of the pyruvic acid produced from the glucose molecule during the glycolysis. The acetyl CoA then enters a cyclic pathway tricarboxylic acid cycle, most commonly as the Krebs cycle, after the scientist Hans Krebs who first illustrated it. फिर आता है ट्राइकार्बोक्सिलिक एसिड साइकिल, TCA cycle. The TCA cycle starts with the condensation of the acetyl group with the oxaloacetic acid and butter to yield the citric acid. The reaction is catalyzed by the enzyme citrate synthase and the molecule of the CoA is released. Citrate is then isomerized to the isocitrate. It is followed by the 2 successive steps of the decarboxylation. कहता है कि it is followed by the 2 successive steps of the decarboxylation lead to the formation of the alpha-ketoglutaric acid and then the succinyl CoA. In the remaining steps of the citric acid cycle, the succinyl CoA is oxidized to the OAF, following the cycle to continue. Then the conversion of the succinyl CoA to the succinic acid and a molecule of the GTP is synthesized. This is a substrate of phosphorylation SLP. In a coupled reaction, GTP is converted to the GDP with the simultaneous synthesis of the ATP from the ADP. Also, there are 3 points in the cycle where the NAD+ is reduced to the NADH plus H+, and 1 point where FAD+ is reduced to the FADH2. The continued oxidation of the acetyl CoA by the TCA cycle requires the continued replacement of the oxaloacetic acid, the first member of the cycle. In addition, it also requires the regeneration of the NAD+ and the FAD+ from the NADH and the FADH2, respectively. The summary equation for this phase of the respiration may be written as the follow. तो कहता है इसमें कि पायरुविक एसिड प्लस 4 NAD+ प्लस FAD+ प्लस 2 H2O प्लस ADP प्लस इनऑर्गेनिक फॉस्फेट माइटोकॉन्ड्रिया मैट्रिक्स में जाता है और बनाता है क्या, तो और बनाता है इसमें है जो 3 molecule of the CO2 प्लस 4 molecule of the NADH प्लस 4 molecule of H+ प्लस FADH2 प्लस द ATP है जो बनाता है. फिर ये दिया हुआ है द सिट्रिक एसिड साइकिल का स्ट्रक्चर. सिट्रिक एसिड साइकिल है जो दिए हुए है द सिट्रिक एसिड साइकिल. इसमें क्या हो गया कि पायरुवेट है जो acetyl CoA के साथ मिलकर CoA के साथ मिलकर acetyl coenzyme बनाया. फिर acetyl CoA जो है oxaloacetic के साथ मिलकर सिट्रिक एसिड बनाया. सिट्रिक एसिड को अल्फा कीटो डायरेक्ट दे दिया, यहाँ पर 5 कार्बन का अल्फा कीटोग्लूटारिक एसिड में कन्वर्ट किए, एक CO2 निकल गया और NADP है जो NADH में कन्वर्ट हो गया. फिर अल्फा कीटोग्रेट मतलब ये डायरेक्ट लिख दिया है, सल्फोनिक एसिड बना दिया है, सल्फोनिक से फिर मैलिक एसिड बना दिया है. ये है जो डायरेक्ट लिख दिया है इस तरह से. फिर आता है कि we have till now seen that glucose has been broken down to release the CO2 and the 8 molecule of the NADH plus H plus 2 molecule of 2 of the FADH2 have been synthesized besides just the 2 molecule of the ATP in the TCA cycle. You may be wondering why we have been discussing respiration at all. Neither O2 has come into the picture nor the promised large number of the ATP has yet been synthesized. Also, what is the role of the NADH plus H plus and the FADH2 that we synthesized? Let us understand the role of the O2 in the respiration and how the ATP is synthesized. उसके बाद फिर आता है इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम एंड द ऑक्सीडेटिव फॉस्फोरिलेशन. द फॉलोइंग स्टेप्स इन द रेस्पिरेटरी प्रोसेस आर टू रिलीज एंड यूटिलाइज द एनर्जी स्टोर्ड इन द NADH plus H plus and the FADH2. This is accomplished when they are oxidized through the electron transport system and the electron are passed on to the O2, resulting in the formation of the H2O. The metabolic pathway through which the electron passes from the one carrier to the another is called the electron transport system. It is present in the inner matrixular membrane. Electrons from the NADH produced in the mitochondrial matrix during the fatty acid cycle are oxidized by an NADH dehydrogenase complex 1 and electrons are then transferred to the ubiquinone located within the inner membrane. Ubiquinol also received a reducing equivalent via the FADH2 complex 2 that is generated during the oxidation of the succinate in the fatty acid cycle. The reduced ubiquinol is then oxidized with the transfer of the electrons to the cytochrome C by the cytochrome B1 complex 3. Cytochrome C is a small protein attached to the outer surface of the inner membrane and acts as a mobile carrier for the transfer of the electrons between the complex 3 and the complex 4. Complex 4 refers to the cytochrome C oxidase complex containing A and the A3 and the two copper center. When the electrons pass from the one carrier to another by the complex 1 to 4, 5 in the electron transport chain, they are coupled to the ATP synthase complex 5 for the production of the ATP from the ADP and the inorganic phosphate. The number of the ATP molecules synthesized depends on the nature of the electron donor. Oxidation of the one molecule of the NADH gives rise to the 3 molecule of the ATP, while that of the 1 molecule of the FADH2 produces 2 molecule of the ATP. Although the aerobic process of the respiration takes place only in the presence of the oxygen, the role of the oxygen is limited to the terminal stage of the process. Yet the presence of the oxygen is vital since it drives the whole process by removing hydrogen from the system. Oxygen acts as the final hydrogen acceptor, unlike the phosphorylation where it is the light energy that is utilized for the production of the proton gradient required for the phosphorylation. In respiration, it is the energy of the oxidation reduction utilized for the same process. It is for this reason that the process is called as the oxidative phosphorylation. फिर कहता है कि you have already studied about the mechanism of the membrane linked ATP synthesis as explained by the chemoosmotic apparatus in the earlier chapter. As mentioned earlier, the energy released during the electron transport system is utilized in the synthesizing ATP with the help of the ATP synthase complex 5. This complex consists of two major components, F1 and the F0. The F1 headpiece is a peripheral membrane protein complex and contains the 5% synthase of the ATP from the ADP and the inorganic phosphate. F0 is an integral membrane protein complex that forms the channel through which the protein cross the inner membrane. The passage of the proton through the channel is coupled to the catalytic site of the F1 component for the production of the ATP. For the each ATP produced, 4H plus passes through the F0 from the intermembranous space to the matrix down the electrochemical proton gradient. तो ये दिया हुआ है इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम ETS का है जो दिए हुए है. इधर है जो कॉम्प्लेक्स 1 से हम FMN है, इसमें से कॉम्प्लेक्स 1 से FMN से इलेक्ट्रॉन पास किया NADH से NAD में है जो ऑक्सीडाइज हुआ, इसे FNN से पास किया, फिर FES से पास किया. FMN है जो हाइड्रोजन कैरियर भी है और है जो इलेक्ट्रॉन कैरियर भी है. फिर FES है जो यूबिक्विनोन को दिया, यूबिक्विनोन है जो हाइड्रोजन कैरियर है तो 2 इलेक्ट्रॉन इधर भी फेंकेगा. उसके बाद फिर आता है यूबिक्विनोन साइटोक्रोम कॉम्प्लेक्स 3 को देता है. कॉम्प्लेक्स 3 में साइटोक्रोम बी के पास पहले इलेक्ट्रॉन जाता है, फिर FES के पास, फिर साइटोक्रोम C1 के पास, फिर साइटोक्रोम C को देता है. साइटोक्रोम C है जो फिर जाके कॉम्प्लेक्स 4 को देता है. कॉम्प्लेक्स 4 में साइटोक्रोम A आता है, साइटोक्रोम A3 आता है. साइटोक्रोम A3 में है जो कॉपर होता है और साइटोक्रोम फिर A3 दे देता है O2 को. O2 है जो H2O बना देता है. ये यहाँ पर दिए हुए कॉम्प्लेक्स 2 जिसको सक्सिनेट डिहाइड्रोजनेज कॉम्प्लेक्स भी कहते हैं. ये जो है FAD को FADH2 में FADH2 को FAD में प्लस में FAD में कन्वर्ट करता है और इसमें है जो ये भी अपना इलेक्ट्रॉन यूबिक्विनोन को देता है और यूबिक्विनोल है जो फिर दे देता है कॉम्प्लेक्स 3 को. उसके बाद फिर दिए हुए डायग्रामेटिक रिप्रेजेंटेशन दिए हुए ऑफ द डायग्रामेटिक रिप्रेजेंटेशन ऑफ द ATP सिंथेसिस इन द माइटोकॉन्ड्रिया दिए हुए कि कैसे ATP सिंथेसिस होता है. तो ये हो गया F0 और ये हो गया F1. F0 जो है वो इंटीग्रल है, F0 जो है पेरिफेरल है. F0 जब H प्लस से पास करता है तो इसमें है जो कॉन्फॉर्मेशनल चेंज होता है F1 में, जिससे ADP और इनऑर्गेनिक फॉस्फेट मिलकर है जो ATP है जो बन जाता है. फिर आता है द रेस्पिरेटरी बैलेंस शीट के बारे में क्या कहते हैं एनसीईआरटी. तो एनसीईआरटी कहता है इट इज़ पॉसिबल टू मेक द कैलकुलेशन ऑफ द नेट गेन ऑफ द ATP फॉर द एवरी ग्लूकोज मॉलिक्यूल ऑक्सीडाइज, बट इन रियलिटी दिस कैन ओनली ए थियोरेटिकल एक्सरसाइज. This calculation can be made only on the certain assumption that there is a sequential order pathway following with one substrate forming the next and with the glycolysis, TCA cycle and ATS pathway following one after another. The NADH synthesized in the glycolysis is transferred into the mitochondria and undergoes the oxidative phosphorylation. फिर आता है none of the intermediates in the pathway are utilized to synthesize any other compound. फिर कहता है कि only glucose is being respired, no other alternative substrate are entering in the pathway at any of the intermediary stage. फिर कहता है कि but this kind of the assumption are not really valid in the living system. All pathways work simultaneously and do not take place one after the another. substrate enter the pathway and are withdrawn from it as they when the necessary ATP utilized as and when needed. Enzymatic rates are controlled by the multiple means, yet it is useful to do this exercise to appreciate the beauty and the efficiency of the living system in the extraction and the storing energy. Hence, there can be a net gain of the 38 ATP molecule during the aerobic respiration of the 1 molecule of the glucose. फिर कहता है कि now let us compare the fermentation and the aerobic respiration. Fermentation accounts for only a partial breakdown of the glucose, where in the aerobic respiration it is completely degraded to the CO2 and the H2O. In the fermentation, there is a net gain of only 2 molecules of the ATP for the each molecule of the glucose degraded to the pyruvic acid, whereas many more molecules of ATP are generated under the aerobic condition. फिर कहता है कि NADH is oxidized to the NAD plus rather slowly in the fermentation. However, the reaction is very vigorous in the case of the aerobic respiration. ये आ गया एम्फीबोलिक पाथवे. The glucose is the favorite substrate for the respiration. All carbohydrates are usually first converted into the glucose before they are used for the respiration. Other substrates can also be respired, has been mentioned in the earlier, but then do not enter the respiratory pathway at the first step. See figure to points of the entry of different substrate in the respiratory pathway. Fat should not be broken down into the glycerol and the fatty acid. First, if the fatty acid were to be respired, they would be first degraded to the acetyl CoA and enter the pathway. Glycerol would enter the pathway after being converted to PGAL. The proteins would be degraded by the proteases and individual amino acid after deamination, depending on their structure. Depending on the structure, would enter the pathway at some stage within the Krebs cycle or even as a pyruvate or the acetyl CoA. फिर आता है कि since the respiration involves the breakdown of the substrate, the respiratory process has traditionally been considered a catabolic process and the respiratory pathway as a catabolic pathway. But is this understanding correct? We have discussed above at which points in the respiratory pathway different substrate would enter if they were to be respired and use the energy, derive energy. But it is important to recognize that it is this very compounds that would be withdrawn from the respiratory pathway for the synthesis of the said substrate. Hence, fatty acid would be broken down to acetyl CoA before entering the respiratory pathway when it is used as a substrate. But when the organism needs to synthesize fatty acid, acetyl CoA would be drawn from the respiratory pathway for it. Hence, the respiratory pathway comes into the picture both during the breakdown and the synthesis of the fatty acid. Similarly, during the breakdown and the synthesis of the protein too, respiratory intermediates form the link. Breaking down the process within the living organism is catabolism and synthesis is the anabolism. Because the respiratory pathway is involved in the both anabolism and catabolism, it would hence be better to consider the respiratory pathway as the amphibolic rather than an catabolic one. तो ये दिया हुआ है interrelationship among the metabolic pathway showing the respiration mediated breakdown of the different organic molecules to the CO2 and the H2O में. तो ये फैट्स हो गया, फैट्स को फैटी एसिड इन ग्लूकोज में तोड़ते हैं. फिर उसके बाद कार्बोहाइड्रेट्स हो गया, सिंपल शुगर ग्लूकोज हो गया, ग्लूकोज 6 फॉस्फेट में तोड़ते हैं, फिर फ्रुक्टोज 6 फॉस्फेट में तोड़ते हैं. फैटी एसिड ग्लूकोज से ढाप बना सकते हैं, फिर ढाप ग्लिसरॉल 3 फॉस्फेट से भी ढाप को है जो ग्लिसरॉल यानी पीजीएल में कन्वर्ट कर सकते हैं और पीजीएल से पायरुविक एसिड से फिफ्टाइल को बना के क्रेब साइकिल में एंटर कर सकते हैं. और अगर प्रोटीन है तो प्रोटीन से अमीनो एसिड में तोड़ेंगे और अमीनो एसिड को पायरुविक एसिड में ब्रेक करके फिर उसको है जो फिटाइल को बना के आगे है जो क्रेब साइकिल में है जो इंटर करा सकते हैं. फिर आता है रेस्पिरेटरी कोफेंट. तो लेट अस now look at another aspect of the respiration. As you know, during the aerobic respiration, O2 is consumed and the CO2 is released. The ratio of the volume of the CO2 evolved to that of the O2. Volume of the O2 consumed in the respiration is called as the respiratory quotient or the respiratory ratio. इसका है respiratory quotient बराबर volume of the CO2 evolved but the volume of the O2 consumed होता है. The respiratory quotient depends upon the type of the respiratory substrate used during the respiration. When the carbohydrates are used as a substrate and are completely oxidized, the respiratory quotient will be 1 because equal amount of the CO2 and the O2 are evolved and consumed, respectively H1 in the equation given below. तो ये आ गया C6H12O6 प्लस 6CO2 6CO2 प्लस 6H2 प्लस एनर्जी आता है. रेस्पिरेटरी कोफेंट आ जाएगा इसका. रेस्पिरेटरी कोफेंट कितना आ जाएगा तो 6CO2 6O2 1 आ जाएगा. फिर आता है when fats are used in the respiration, the RQ is less than 1. Calculation of for fatty acid tripalmitin if used as a substrate is shown below. तो कहता है कि ये ट्राइपैल्मिटिन है, इसमें है जो O2 डालें तो है जो रेस्पिरेटरी कोफेंट कितना आता है. ट्राइपैल्मिटिन के लिए रेस्पिरेटरी कोफेंट जो आता है 0.7 आता है. ये ट्राइपैल्मिटिन है, इसका रेस्पिरेटरी कोफेंट है जो 0.7 है. उसी तरह से कार्बोहाइड्रेट का रेस्पिरेटरी कोफेंट कितना आता है तो 1 आता है. तो ये मेन है. प्रोटीन रेस्पिरेटरी सबस्ट्रेट द रेशियो वुड भी 0.9 आएगा. बट इंपॉर्टेंट टू रिकॉग्नाइज दैट लिविंग ऑर्गेनिज्म in the living organism respiratory substrate are often more than one प्योर प्रोटीन्स और फैट्स आर नेवर यूज्ड एज ए रेस्पिरेटरी सबस्ट्रेट के रूप में। प्योर प्रोटीन्स और फैट्स को हम कहीं भी रेस्पिरेटरी सबस्ट्रेट के रूप में यूज नहीं करते हैं। फिर आता है कि प्लांट समरी इसका है कि प्लांट्स अनलाइक एनिमल्स हैव नो स्पेशल सिस्टम फॉर द ब्रीथिंग और द गैसियस एक्सचेंज ऑफ टोमाटा एंड द lentil cells allow the gaseous exchange by the diffusion. Almost all living cells in a plant have their surface exposed to the air. The breaking of the CC bonds of a complex organic molecule by the oxidation cell leading to the release of a lot of the energy is called as the cellular respiration. Glucose is the favorite substrate for the respiration. Fats and the proteins can also be broken down to yield energy. The initial stage of the cellular respiration takes place in the cytoplasm. Each glucose molecule is broken through a series of the enzyme catalyzed reaction into the two molecules of the pyruvic acid. This process is called as the glycolysis. The fate of the pyruvate depends upon the availability of the oxygen and the organism. Under the anaerobic condition, either lactic acid fermentation or the alcohol fermentation occurs. Fermentation takes place under the anaerobic condition in many prokaryotes, unicellular eukaryotes, and in germinating seeds. In the eukaryotic organism, aerobic respiration occurs in the presence of the oxygen. Pyruvic acid is transported into the mitochondria where it is converted into the acetyl-CoA with the release of the CO2. Acetyl-CoA then enters the thiocarboxylic acid pathway or the Krebs cycle operating in the matrix of the mitochondria. NADH plus H plus and FADH2 are generated in the Krebs cycle. The energy in these molecules is like that in the NADH plus H plus synthesized through the glycolysis are used to synthesize the ATP. This is accomplished through a system of the electron carriers called the electron transport system located on the inner membrane of the mitochondria. The electrons as they move through the system release enough energy that are trapped to synthesize the ATP. This is called as the oxidative phosphorylation. In this process, O2 is the ultimate acceptor of the electrons and it is referred to the barter. The respiratory pathway is an amphobulic pathway as it involves both the anabolism and the catabolism. The respiratory quotient depends upon the type of the respiratory substance used during the respiration.